the secondary structure of tRNA has been depicted that looks like a clover leaf-like model. Its actual structure, the tRNA, is a compact molecule which looks like an inverted L. So initiator codon AUG, which is present on the mRNA. So we know, right, which is the initiator codon AUG. Now the large subunit of ribosome joins with the smaller subunit that requires magnesium ions. Enzyme ribozyme acts as a catalyst that brings about the addition of the amino acid to form a long chain. At this time, the first tRNA at the P site is kicked off. Hello everyone, a warm welcome to the session for class 12 CBSE Biology. I am Dr. Divya, Biology faculty. So in this session for chapter 5, molecular basis of inheritance, we will learn about translation mechanism. So in the previous session, that is in session number 9, we had learned about transcription mechanism. So in this session, we'll concentrate on translation mechanism. So we know that the mRNA strand that will be formed, that is during the process of transcription, that will contain the complementary codes that were present on the coding strand of the DNA, and it will, the nascent mRNA that is there, it will fall out of the DNA strand, and later on it will undergo a mechanism called as translation. So transcription was the messages that was there on the DNA, they get copied or transcribed onto the mRNA strand. Now the transcribed gene sequences that are there, those sequences which will be called a gene in the form of triplet codes, right? So those triplet codes, they code for a particular amino acid. Now they have to get translated into amino acids, finally leading to the formation of protein. So how exactly it will happen, we will look in this particular session. So before moving to that, that is the actual process of translation, you need to know about an important RNA molecule which is called as the tRNA. So tRNA. So tRNA is nothing but transfer RNA. Transfer RNA. So like how we have the messenger RNA which carries the messages of the DNA on its strand, that is messenger RNA. Now those messages needs to be translated into amino acids and then into protein. So that is the transfer RNA. So this transfer RNA has an anticodon loop. So knowing the structure of the transfer RNA is quite very important here. So the codon loop, it is present on the mRNA strand. So on the codon loop, there will be the start codons, there will be the stop codons, and in between there will be the gene sequences that are there or the genetic codes that are there, it will be there. So here on the codon, so this is the codon strand, that is the mRNA, it is the codon strand which runs from the 5' to the 3' direction, and in the tRNA molecule it needs to have a strand that is complementary to the mRNA strand, and that strand we call it as anticodon. Anticodon means which is opposite to the codon strand. So therefore meaning opposite to the codon strand means in the codon strand, if the nitrogenous base is adenine, then here it will be uracil on the tRNA. So this is the tRNA. And next, if it is guanine on the mRNA, then on the tRNA strand, it will be cytosine. Similarly, if it is uracil on the mRNA strand, it will be adenine. So, there are different ribosomes which are produced, which will have anticodon sites in them. So, that is one of the region of the tRNA. So, therefore, only that tRNA which has the anticodon to a particular codon will come and bind here. Similarly, if you can see here, UAC, for UAC, obviously AUG, the tRNA having an anticodon, AUG will come and bind. Or say for example, it is UGA. For UGA, so UGA is present on the mRNA. Now, which is the anticodon tRNA that will come and bind here? It will be ACU, right? So, it is like that. So, the tRNA, it will be ACU. So, therefore, it will, those tRNA which is having the respective complementary sequence or the anticodon to that particular mRNA sequence will come and bind here. So, therefore, tRNA has an anticodon loop. We call it as an anticodon loop because doesn't this portion look like a loop? What do you mean by loop? This is a loop. So, this is the first loop, the second loop. So, these curls that are there, we call them the loop. So tRNA has an anticodon loop that has bases complementary to the code, and it also has amino acid acceptor end. So it has an amino acid acceptor end. So when I say amino acid acceptor end means it is that end which accepts the amino acid. Say for example, this is UCA. UCA, if it codes for serine, so that it will have an amino acid acceptor end which is present towards the 3' end. Just like how the DNA and the RNA has 5' 3' ends or 3' 5' ends, same way this tRNA will also have a 5' and a 3' end. So towards the 3' end, it will have a amino acid acceptor region. Amino acid acceptor region. So in this amino acid acceptor region, it will accept what? Serine. Why does it accept serine? Because UCA that is there here, it codes for serine. So that is how. So it will accept serine. Next, moving further, if you can see here it will accept tyrosine because the anticodon strand is AUG, which is complementing to UAC, and AUG codes for the amino acid tyrosine. So these are the amino acid acceptor. And so till now, what did we get to know? What are the parts of the tRNA? So the tRNA has a loop. In that loop, there is the anticodon region that is present that codes complementary to the sequences present on the mRNA strand. Apart from that, on the 3' end of the tRNA is present the amino acid binding site. So much we know. Now, tRNAs, they are specific for each amino acid. That is why, because this region has, this is anticodon for AGU, this is anticodon for UAC. So whatever is there, or say for example, as I told you, here if it is UGA is the sequence here, then which one will come and bind here? It will be A, C and U. So therefore, that tRNA will come and bind here, right? And therefore, ACU will code for a particular amino acid. So that is how. So each tRNA molecule that is there will code for only one particular amino acid depending on the anticodon that is present on its strand or present on the tRNA loop. And for initiation, there is another specific tRNA that is referred to as initiator tRNA. So initiator, so we had learnt in the previous session, that is on the mRNA strand or on the DNA strand also, there are initiator codons that are present and there are terminator codons. So AGU is a, for example, this AGU is the initiator codon. Initiator codon. So if you don't know about it, you can go to the previous session wherein we learnt about genetic code. Have a look at it. And UAC, UGA, UAC, UAG, all these are the stop codons. So what do we mean by initiator codon and stop codon? So the coding for the amino acid begins at the initiator codon and it ends wherever the stop codon is there. And the stop codon, they do not actually code for any amino acid, or there might be a stop codon that is UAA, UAG and UGA. So until the stop codons, remember, they do not code for any amino acid. So until it reaches the stop codon. So UAC is not a stop codon because it codes for the amino acid tyrosine. So UAA, until it reaches this, it will, addition of the amino acid or the arrival of the tRNA on the strand will keep on continuing for a particular, to code for a particular amino acid. So therefore, the secondary structure of tRNA has been depicted that looks like a clover leaf-like model. Its actual structure, the tRNA is a compact molecule which looks like an inverted L. So tRNA, they have given different shapes for it. So it can be clover leaf model, but actually it looks like a L. So actually, if you have to write in the exam what is the shape of the tRNA, you can write it as it looks like a clover leaf. So this is about the tRNA. So we got to know about the tRNA. It is the adapter molecule because it adapts itself on the mRNA strand to bring the amino acid, respective amino acids, and therefore it forms chains of amino acids. So here chains of amino acid will get formed. So serine may be next to valine and next to it will be leucine. Like it forms chain, and each amino acid is held together by peptide bonds. We'll talk about that later. So the amino acids are held together by peptide bonds, and therefore a chain of amino acid is nothing but one particular protein. So that is how protein gets produced. So this is about the tRNA, the adapter molecule. So moving further, we'll talk about the mechanism of translation. So in translation, there is different steps that are involved. So the first step that we'll talk is initiation. Initiation means initiating something or starting something. So initiation here is the starting, wherein the production of the amino acid will start, wherein the tRNA will go and bind. So the activation of amino acid is essential before translation starts. So amino acid is activated. So amino acids, they need to get activated as well as and when the tRNA brings the amino acid and it binds there, the amino acid needs to form a chain. So if they needs to form a chain, they need to have bonds in between. So to formation, that is the peptide bond. So for the formation of the peptide bonds also, a lot of energy is required. So all this first itself, it should be ready before the translation mechanism begins. So amino acid is activated by utilizing energy. I told you a lot of energy is needed, and that energy is got from the ATP, that is adenosine triphosphate. And this amino acid, it binds with the amino acid binding site of the tRNA. I have explained it all this in the previous slide itself. And forms of tRNA amino acid complex. So what will happen here? So the tRNA, when it codes, so on the tRNA, there is the anticodon loop that is present. The anticodon loop, which is complementary to the sequences present on the mRNA strand. Once it comes and binds here, say for example here, this is the codon here. So for GCA, which is complementary, it is CGU, right? So wherever the tRNA that is having the anticodon CGU, it will come and bind here. So when it comes and binds, now along with that, ribosomes also will come and bind, because protein needs to get formed from the chains of amino acid. We'll talk about that later. So these will come and bind. So when they bind, what will happen? For all this, a lot of energy is needed. For that energy, it is utilized from the ATP. So a small subunit of ribosome attaches to the mRNA at the 5' end. So towards the 5' end, a small subunit of ribosome. So you have learnt about ribosome. Ribosomes are nothing but the organelles that synthesize proteins, and they're made up of two subunits, one large subunit and a small subunit. They will also be ready because they need to synthesize the protein. So that is why. So initiator codon AUG, which is present on the mRNA. So we know, right, which is the initiator codon AUG, and it initiates the process of protein synthesis. So wherever AUG is there exactly the initiation or the first binding of the ribosome will take place, and after the ribosome binds, the tRNA will come and bind there, which is anticodon, which is complementing the mRNA sequence. So for AUG it is there means which will come and bind. So first always the complementary tRNA, this is on the mRNA, it is the start codon, right? It is the start codon, and always the translation mechanism should begin at the start codon or initiate at the start codon itself. So for this UAC, the tRNA which is having a complementary strand, UAC will first come and bind, right? So we had seen this in the previous slide. You can have a look. UAC tRNA will come and bind, and it will initiate the process of transcription. So after the initiation of transcription, what will happen? We will look into. So first, what will happen in initiation? All the amino acids that needs to participate in the process of translation should be initiated first. It should be ready. It should be ready wherein the amino acid should be activated by utilizing some of the energies from the ATP molecules that are being produced. So ATP molecules will be utilized, and this amino acid will have an active binding site to which the respective amino acid will come and bind depending on what codon is present on the tRNA. After that, what happened? A small subunit, now it forms a tRNA amino acid complex, right? So after the formation of the tRNA amino acid complex, then a small ribosomal unit will come and attach towards the 5' end. All this will happen in the initiator region itself. That is the AUG sequence or triplet code that is present on the mRNA strand. So then later on the protein synthesis will start. Now initiation has happened. So in the initiation region, ribosome is there. Then comes the tRNA complex, that is the tRNA amino acid complex is there. Everything is there. It is ready. Next, what will happen? We will look into. Next, the initiator charged tRNA. Now it has become an initiator charged tRNA. That is because the tRNA is having a activated amino acid. It's having an activated amino acid methionine because AUG it codes for, or the initiator codon that is there, it actually codes for the amino acid methionine. So the first amino acid that will be formed from the mRNA strand will be methionine, and it binds with the initiator codon, that is AUG, by its anticodon UAC through hydrogen bonds. Remember, the binding between these two, that is AUG is bound, or say for example CGU is bound with GCA with which bond? So these bonds are called as hydrogen bonds. If you remember, A with U and G with C. So these bonds which are there, we call it as what? Hydrogen bonds. Between the nitrogenous bases, the binding happens, or between the complementary nitrogenous bases, the binding is by the hydrogen bonds. Between the amino acids, the binding is by the peptide bonds. Always remember that. Don't get confused with that. And it carries the activated amino acid methionine in the case of eukaryotes. But if the translation mechanism is taking place in the prokaryotes, then the amino acid that will be first formed will be formyl methionine, because it is a different code there. So that is why. And it occupies the P site of the ribosome and the A site is left vacant. So here what will happen? If you can see, the P site it will occupy and the A site will be left vacant. So next, now the large subunit of ribosome joins with the smaller subunit that requires magnesium ion. So for this particular initiation to take place, first tRNA molecule energy is required from ATP, and next we require Mg++ ions as well. So these are the different things that are required for translation mechanism. So moving further, we'll talk about now the initiator region is ready. In the initiator region, ribosome is there. In the initiator region, the activated tRNA amino acid complex is also there. Next is elongation, that is wherever the codes are present, triplet code is present, for each of it complementary tRNA molecule should come and form and it gets elongated. That is what we call as elongation. So during this process of elongation, activated amino acids that are there, they get added one by one. As I told you, say for example, here tRNA which is complementing the GCA will come and bind here. Next tRNA which is complement, next the ribosome will move to the next after that. And as and when, say for example here, it has come and bound to UCC. Which one? The tRNA which is carrying AGG has come and bound here. For UCC it is AGG, right? So it has come and bound here. Now the work is done. The ribosomes will move to the next triplet codon, then to the next triplet codon. Therefore, it will keep on moving, and everywhere when it is moving, say for example here it is AAU. For this it will be UUA. For this it will be UUG. For this AUAG, like that. So for each of these, the tRNA will come and bind, and as and when the tRNA is binding for each of these, say for example for AGG it will produce glycine, next it will produce leucine, next it will produce tyrosine, like that. What type of amino acid is binding, they depend on that. You can have a look at the genetic code that I had taught in the previous session to know which code codes for different amino acids. So this is how they will come and bind. See here, when they are binding, chains of amino acid will get formed. Between these amino acids, they are held by a bond, and that bond we call it as a peptide bond. So this is how elongation process is happening. So like that, for one protein, 20 different types of amino acids will form chain and produce a particular protein. And what that protein is depends on what amino acid chain it is made up of. So that is about this particular elongation. So during this process, activated amino acids get added one by one to the first amino acid. So the first amino acid that will be added will always be methionine, because methionine is the one produced by the tRNA molecule that comes and binds to the initiator codon. So that is why. And the addition of amino acid occurs in 3-step cycle. First is the tRNA should recognize the codon, that is the amino acid tRNA, that molecule that is there. It will enter the ribosome, that is the A site of the ribosome molecule, and the anticodon binds with the codon by hydrogen bonds. I have told you that before itself. Amino acid of the first initiator tRNA, which is there at the P site, and the amino acid on tRNA at the A site, both will be joined by the peptide bond. So, to easily understand, the amino acids keep on forming chain by forming peptide bonds in between. And here the enzyme ribozyme acts as a catalyst that brings about the addition of the amino acid to form a long chain. At this time, the first tRNA at the P site is kicked off. Next, what will happen? The tRNA will be removed, and next, if you can see here, this gets removed. So, each time when the ribosome moves and the function of the tRNA is done, the tRNA is removed, because it should not keep on coding again and again, right? So, that is why once the coding is done, it is just removed off. So, that is how chains of amino acids keep on forming. So, this is about elongation. Next is translocation. So, the tRNA at the A site carrying a dipeptide at A site moves to the P site. So, from one region to the other, it keeps on moving. So, this process is called as translocation. So, in translocation, both the subunit of the ribosomes move along in relation to tRNA and mRNA. So, for the protein synthesis ribosome, that is how it plays a very crucial role. So, the entire ribosome with the larger subunit and the smaller subunit, it keeps on reading the strands along with the tRNA adapter molecule or tRNA amino acid complex. And this process of arrival of tRNA amino acid complex, formation of the peptide bond. So, after the amino acid formation, each amino acid should be linked by the peptide bond, and then ribosomal translocation, that is the ribosome after that particular region, it will move to the other sequence, then to the other sequence. So, ribosomes keep translocating to read the mRNA strand, and removal of the previous tRNA are repeated. So, it keeps on repeating, the same step keeps on repeating. That is initiation and elongation, both these steps keep on repeating until, as well as translocation keeps on repeating until chains of amino acids are formed. And finally, these chains of amino acids will be nothing but the proteins. So, after that, so now chains of amino acids are formed from the initiator region up till the terminator region, or up till where the stop codon is present. Now, when that is done, next what happens we will look into, that is termination and release of polypeptide. So, towards the 3' end of the, so initiator codon is present towards the AGU, is present towards the 5' end, and the terminator codons are present towards the 3' end. So, the terminator codons, which we also called as the stop codon, that is UAA, UAG or UGA, it is exposed at the A site, right? It will be somewhere in the A site, and it is not read or joined by any anticodon because they do not code for any amino acid, and their only function is to stop the coding for the amino acid. And once the stop codon reaches, the release factor binds to the stop codon, and thereby the process of translation, it comes to an end or it will terminate. Then the polypeptide chain, what is that polypeptide chain now? Nothing but chains of amino acids which are there, it will be released into the cytoplasm, and therefore in the cytoplasm we have chains of amino acids with respect to particular proteins, and that is how proteins get produced in a body. So, this is about the termination and release of the polypeptide. So, next after that, 2 subunits of the ribosome. So, now the tRNA got separated, the polypeptide chains got separated. What about the ribosomes which are there on the mRNA strand? That is the larger subunit and the smaller subunit. So, the 2 subunits of the ribosome, they dissociate once they reach the terminator region on the RNA, and they are released back to the cytoplasm. Their work is done and they will go back to the cytoplasm. Next, again translocation mechanism or translation mechanism should happen. Then again these ribosomes will come back to do their work of protein synthesis. That is how ribosomes actually synthesize proteins. Ribosome alone cannot synthesize protein. They synthesize protein by reading the strands that are present on the mRNA. And mRNA also has some additional sequences which are non-translated. Those sequences which we call it as the UTR, that is untranslated region. There are some strands which do not get translated because maybe they might be lying after the terminator region, right? So once it terminates at one particular point, say for example, the reading has happened from here from initiation till terminator. What about this region? Did it get translated? No, right? So this region which did not participate in the formation of amino acid or protein synthesis, or which did not participate in the process of translation, we call it as the untranslated region. Now what happens to the remaining untranslated region? So the untranslated regions that are present both towards the 5' end and also towards the 3' end, that is before the start codon also it will be present, right? Here also we can have some untranslated region. So it can be present. So see here itself, if you can see here itself. So this is the UTR which starts before the initiator. This can also be the UTR which starts after the terminator. So what happens to these untranslated regions? So the untranslated regions that are present, they are required for efficient translation process also. And finally the mRNA is also released in the cytoplasm and it gets denatured by the nucleases immediately. So the untranslated region that is there, that is of no use, right? So it gets degenerated. It will be, the nucleases will just degrade it and it will just vanish off from the cytoplasm, and hence that is why mRNA is always short-lived. So DNA is long-lived, mRNA is short-lived. So mRNA is short-lived because of this particular region. Once translation is done, the untranslated regions of the mRNA, they get denatured or they get degraded by the nucleases enzyme present inside the nucleus, and then the death of the mRNA. So to make you understand, I can say the death of the mRNA strand will occur. So therefore mRNA is short-lived. So this is about the termination and formation of polypeptide chain. So this was about the session wherein you got to know that how the tRNA brings the amino acid and chains of amino acids will get formed, and these chains will just liberate itself and it will go into the cytoplasm and proteins will be synthesized with the help of ribosomes. So all the functions of the ribosome, the tRNA molecule, the initiator codon, the terminator codon, what happens to the untranslated regions of the mRNA, all that you got to know. So this was about this session. So with this, translation mechanism comes to end. So it is a beautiful topic, like what is the function of a DNA? We all know that we have DNA, we have genes. So what exactly these genes do exactly? This is what they keep on doing inside a cell. So all cells are quite active, right? They do not sleep. They continuously keep on synthesizing amino acids and proteins in a body. And they are very, very important for all the activities to take place in a body. So this was about this session. So in the next coming session, we'll talk about regulation of gene expression, how the genes get regulated and how do they get expressed. And in that particular case, some of the genes, they get switched off, they get switched on, and all that we will learn. And to explain regulation of gene expression, we'll take the example of lac operon concept. So lac operon concept is quite important in terms of exam point of view. So this was about this session. So we shall meet in the coming session with these two topics. Thank you.